Boa tarde a todos, boa tarde aos que estão presentes aqui, aos que estão online e aqueles que vão assistir via YouTube futuramente. Hoje nós vamos ter a apresentação do professor Mathieu Javaux, da Universidade Católica de UCLouvain. Ele é especialista em física dos solos, raiz de planta, sistema solo, planta, atmosfera. And Mathieu, the floor is yours.
Thank you very much, Laura. So today what I will try to do is to explain a little bit what I've been doing during my last years. But maybe to start, a few words. How do I change the slide? About me? Okay. So I'm from Belgium. Now? No. So I'm from Belgium. Belgium is a small country over there that you see. And there I'm working at UCLouvain, at Earth and Life Institute. It's a new city in the middle of Belgium, southern Brussels. And I'm also working in Germany, very close to the border, in Juelich. So I have two affiliations. The first in Louvain-la-Neuve, where I did my PhD. It was a PhD on solute transport. Then I moved to Juelich from my postdoc. And then in 2006, I become appointed both in Louvain and in Juelich until now. I first wanted to add a few slides on why would we study vegetation and vegetation transpiration? Why is it so important to study that? And I list several aspects. The first aspect is that 60% of the rainfall that arrives on continents move back to the atmosphere through evapotranspiration. So 60% is a lot. It means that the water cycle, the terrestrial water cycle, the continental water cycle is very much affected by the vegetation. A second reason is that when we investigate in different studies, can we take this out? I don't know. Is there some information there?
One more. Floating meeting.
Okay. So if you try to estimate the amount of actual evapotranspiration, so if you want to know, okay, what is the global evapotranspiration over the Earth, you have a lot of different options. You can do it based on statistical models, you can do it based on remote sensing, you can do it based on isotopes, and all these methods provide very, very different estimations. So actually, you see, if you look at how much actual evaporation you have, it goes between 25% and 90%. So every study has a very different estimate of the amount of water that goes back to atmosphere through plants. So it's another good reason to study transpiration is that it's a very badly estimated variable. The third reason is climate change. This is an estimation of the evolution of the evapotranspiration on Earth in the coming 80 years. And you see that we will have a significant increase. You have different scenarios there. So at least 40 millimeter per year. That's the current projections for the future. And last but not least, it's an interesting aspect. So you have a lot of complexity. On the left, this is an image where you see a root. This is a root with root hairs. So this must be like 500 micrometers, maybe a bit less, 200 micrometers. And you see the distribution of water basically around the root. And here it's a maize field. You have maize rows, and this is the water content in 3D below the maize in the soil. And you see whether you look at micrometer scale, whether you look at centimeter or meter scale, and of course, if you go to continental scale, the distribution of the water is very, very heterogeneous, and it's not easy, it's difficult to measure, so it's fun. There's an additional problem when you study soil plant atmosphere, is the difference of scales that are to be taken into account. You have things happening at very, very small scale, like the movement of water in the soil, in the plant. You have things happening at the profile scale, the root distribution, the soil profile. You have things working at the landscape scale, the location of the trees or the vegetation along the landscape, the groundwater, and other aspects. And then you have the climate effect. All that is mixed. And the problem we have is that the scales of the problems we want to solve are rather these two, regional scale, field scale, global scale. And where we measure is rather at small scale. So we have a kind of scale gap between what we measure and what we want to solve. And that's a big issue in soil-plant interaction. So what I've been doing until now, I've studied several aspects. I've started my career working on pela transfer functions. Then I've been working a little bit on surface runoff, surface connectivity. Then during my postdoc, I started to work with 3D models and root water, trying to investigate the uptake of plant, the transpiration of plant. And lately, I was very much interested by upscaling what we observe at small scale to large scale. So the questions I ask myself when I'm doing my research are these ones. So the first is, how does vegetation transpiration react to either soil or atmospheric growth? How can we represent these processes at different scales? So I know that roots are taking up water, that there are a lot of different roots, that you have fields with different types of vegetation, and the upper your scale, the more complex is the system, but is there a way to take into account this diversity, this heterogeneity? And when we speak about heterogeneity, the question is, which heterogeneity matters? So what should we take into account? Is it the heterogeneity of soil, of vegetation, of weather? Which property, trait, or factor is relevant to predict large-scale transpiration flows? And how can we characterize these factors? The other question is related to root and soil hydraulic properties. I'm working a lot with hydraulic properties of soil and then of roots, and how can we characterize them? It's not so easy. And then there is also the long-term reaction of vegetation to drought. If a plant, if there is a drought affecting plants, vegetation reacts in closing stomata, of course, but you have also medium-term reaction, what we call plasticity. So where do you allocate the carbon? How do you adapt to your climate, to your environment? That's also important to consider. And I have several hypotheses in what I'm doing, and I've always followed them since the beginning. First, I believe that physics can help, that the physical laws can be applied at any scales, and that as long as we properly define the variable of interest, we can always apply very simple physical law. I also know that the scale at which the processes occur are not always the scale at which they have an important effect. You know that transpiration is happening through a very tiny hole at the surface of the leaves. It's a very small process, but it affects global water fluxes. And what I'm always trying to do is to combine experiments and models and try to capture the key processes to extrapolate or scale up at larger scales. So now, water flux in the SPAC. You know what is the SPAC? SPAC is soil, plant, atmosphere continuum. So this is physical understanding of the water flow from the soil towards the atmosphere. So the water flow is following a gradient of potential. In the air, you have a water potential of around 100 megapascal, in the soil of minus 0.1 megapascal. The potential is lower in the atmosphere, so the water is sucked up from the bulk soil to the atmosphere. And in its pathway between the subsoil, the soil or the bulk soil and the atmosphere, the water encounters a series of resistances. So there is the soil resistance, or the rhizosphere resistance, the xylem, resistance, the root resistance, and probably the biggest resistance lies in the stomata. You know, plant can open and close the stomata, and this is a very important resistance. If you make this capillary hypothesis or electricity comparison, you can compare this to an electric circuit where you have different levels of potential, and between these levels of potential, you have resistances that control the intensity of the current. So it's a very simple law that we can apply here. If we go a little bit further, you all know, because you have a garden, you have plants, that when the soil dries out, the plant wilts, so the stomata close. And you also know that when the air is very dry, the stomata close as well. And there is a debate about what is the main driver. Is it rather the air, rather the soil, and how does the plant react to these two demands or constraints? So if we go into a bit more detail, we can look at all these different resistances. So sometimes I say resistance, sometimes I say conductance. Conductance is the opposite of resistance, and vice versa. So stomatal conductance, you know, these tiny holes, they regulate transpiration. They depend on the pressure in the leaf. They depend on ABA, on a lot of different processes. Xylem conductance, xylem is this tissue within plants where the water is passing through. Well, the size of the xylem vessels, the connection between xylem vessels affect how easy it is for plants to take up water and to transport water. Something that happens in xylem is embolism, also called cavitation, that drastically decrease the conductance of the xylem. You have the root. Roots are very complex systems where you have different types of tissue. This is a cross-section of a root. Roots are continuously evolving, growing, going deeper, going towards water. They shrink, they swell, and they change in terms of conductance as well. And then between soil and root, you also have another resistance that we will call rhizosphere conductance. In stomata, we can, or people do, compare how the stomatal conductance, here it's called GS, stomatal conductivity, stomatal conductance, is related with the potential. Because perhaps I didn't mention it. I don't know if I can go back. Anterior, yes. But you also have potentials. This side here are potential levels. Potential in the soil, in the soil-root interface, in the xylem of the leaf, and in the atmosphere. And so you can actually make these conductances function of the potential. So for instance, this is a review by Underhead where it looked at very different species, gymnosperms, angiosperms, tropical species, tropical evergreen species, and it looked how the stomatal conductance was a function of the potential, of the pressure, of the negative pressure. And you see that you have a certain shape, a decreasing shape. If you have a potential very close to zero, it's not very negative, you have a maximum conductance, and if the potential drops, stomata close. You have the same in the xylem. So if the xylem cavitate, cavitation means bubble appearing in the xylem. This appears when you have very negative potential. This is a bit complicated axis. Instead of putting the total conductivity, they put the loss of conductivity. So when there is zero loss, it means the conductance of conductivity is maximum. When there is 100% loss, it means that the conductance is zero. And here, so you see a function of the water potential, of the xylem water potential. Where the xylem water potential is close to zero, everything is functioning properly. And if you have a very negative potential, cavitation appears and you have a loss of conductivity. In the root as well, you have a lot of complexity. This is a cross-section of a root at different locations. This is the tip of a root segment. You can imagine that water has to cross this root, so you have different layers of cells. You have the exodermis, you have the endodermis. This is the xylem vessels. You have the cortex, and water is moving there. And each of these tissues has a different conductivity, a different resistance to water. Plus, the shape of the root system will also affect conductance. So all these tissues, all these pathways, potential pathways for water, have their conductivity. In soil as well, you have resistance or conductance or conductivity. I will come back to this very quickly, but what we call the rhizosphere conductance is the fact that if you have a root, around the root, you have a drop of water content or of water potential that generates a high resistance. So when the soil is dry, you have a large resistance around the roots that impede the water movement of the water towards the soil. And there, maybe what I should do is a couple of slides about some properties of soil, because this is my background. Soil is a very complex system. You have different size of grains, and in soil, typically, we consider that there are two main driving forces: the capillarity and the gravity. Capillarity is very important in dry soil, gravity more in wet soils. We always speak in terms of potential. So I've already mentioned a lot of times potential, calling water potential. Usually we explain it either in Pascal or in water head in centimeters. Behind that, it's an energy level. And in soil, we have a special component of the water potential, which is called the suction head. Suction head is the energy, the potential energy due to capillarity. And to characterize soil, we have two characteristic curves. One that relates the water content with the level of energy is called retention curve or PF curve, and each soil type has a different curve. Here you have a clay, a loam, a sand. The relationship is very characteristic for given texture. And the other one is what we call conductivity curve, where you see for different types of soil, different relationship between the conductivity, the ability to transfer water, and again, the potential or the suction. So the soil also has this K curve, this conductivity curve. And of course, if you remember of your electricity lectures, you know that if you have conductivities or conductances in series, this is the minimal one that controls the full flux. And the question is, at the end of the day, where is the minimal conductivity? Here, it was a review where we put the conductivity observed in biological science, they are summarized there, versus the conductivity of soils in purple. And what you see basically is that if the soil is wet, if the water potential is close to zero, soil is much more conductive than roots. In green, you have the bar, the envelope of all the measurements made by biologists. If the soil is rather dry, in this range, I think there is a, so in this range, you see that the conductivity of roots in green are much higher. It should be much higher than the conductivity of soil. So when the soil is dry, soil is limiting the flow. When the soil is wet, root is limiting the flow. So what I'm going to do now is to give you different ways, different studies that we show you, different studies in which we try to predict transpiration, predict root water uptake. We will go from very detailed 3D approaches to a global study at the global scale. This is the only equation of the talk. This is Richard's equation for soil physicists. It's a very known equation. It's very known, but not so easy to solve. It's you have two characteristic curves that I just explained. KH, the conductivity curve, the conductivity function of the soil, and behind CH you have the retention curve of the soil. And these two curves are highly nonlinear. So when you want to solve it numerically, it's not that easy. And you have an S there, and what the S there is what we call the sink term, is the uptake of water by plants. And the question is, how do we model plants? How do we model this S? The first approach that has been used in literature is to use an explicit representation of the plant. It means that we want to represent the plant with each of its root segments. So we created a model where we can represent each of these roots. We solve the equation within the root, within the soil. We have a demand, an evaporated demand. We have water in the soil, heterogeneously distributed. We have different radial, axial parameters, xylem parameters in the plant, and we can solve the system. There was a beautiful video here where you could see that root was growing and water content was changing. So, and you could see the heartbeat of the soil with day-night uptake. So you can model water potential, water content, and evolution of plant in 3D. With this kind of model, you can do a lot of things. You can do pesticide uptake, you can do signaling in the root, like these split root experiment. You can model mucilage. You can also model how roots find macropores and then go down. So you can do really a lot of things, and this model has been developed for several years to try to improve all these processes. But there is another approach, because of course we don't have always, and it's very rare, to have access to the full root system. So in most of the questions, what we have access to is just some values. And then the question is, can we characterize a plant without knowing the full root system? How do we do it? What we can do is to find root system effective property. And this, we did it. I forgot the equation. It's a bit complicated, but basically what you have to know on this slide is that we can define 3 parameters or 2 parameters, one which is related to the root conductance and one which is related to the root distribution. And these 2 parameters are exactly or contain exactly the same information than a full 3D root system. So mathematically, we can prove that the way how we represent the root system in 1D or 0D or 2D is as precise as if you would have the 3D root system. So this was mathematical type of development. And with this type of development, we can see the link between what biologists call root traits. For instance, ABA, you know that ABA can open and close aquaporins. Aquaporins are those tiny proteins that allow water to enter cells. So you can play with the aquaporin activity. You can also have plants which create gaps between the soil and the root. You can also have plants which have high aquaporin activity and gaps. And you can see how, if you have this type of plants, they differ in terms of transpiration. So if you have gaps, you have stress a bit earlier. If you have high aquaporin in blue activity, you have this blue curve with a stress which arrives later. So yeah, we are able to represent. And we are also able to represent, based on this very simplified model, the conductance of a plant. Here what we did, it was for maize or corn. What we try to do is try to estimate what is the root conductance, so the ability of the root system to take up water, at different age of a maize root, from the seed down to 90 days old, where the root system doesn't grow anymore. And just based on simple information, like the age, we were able to represent with Marshall, the model we developed, the envelope of all the observations that were observed through the. Okay, this is nice. We're able to predict things. We're able to run models very complicated, but can we do something more simple? I say simpler, but maybe it's not so simple. Here, what we did is to say, okay, let's simplify the system with 3 conductances in series. Let's solve it. And let's solve it in 0D, so no dimensional aspect. We have 3 equations: Gardner model, Couvreur model, and a model of transport in the xylem put together. And then we look at the relationship between transpiration, E is transpiration here, the soil water potential, so its ability to attract water, or retain water, sorry, and here the leaf water potential. So what we see in front of us is the relationship between how much a plant can transpire as a function of the soil water potential and the leaf water potential. Another way to look at it is to say, if I want to transpire this value, and if the soil is at that level, then the leaf water potential should be that. So this is a physical representation of a plant soil system at a given moment. And it's just physics. And what we see is this shape. This is not a volume, this is a surface that represents the relationship between these three values. And behind that, you have only a kind of Ohm equation, except that K, the conductivity, the conductance of the system, is not constant. If K was a constant, this would be a flat linear plane. Because K is not constant, then you have a strange shape. And this strange shape, yes, I need to explain a few things. First, very easy part of the curve. If the transpiration is 0, it means that the soil and the water potential are equal. If Psi leaf is Psi soil, then E is equal to 0. So this is an easy part of the equation. This equation is when Psi soil is 0. When Psi soil is 0, then K is the conductance of the plant, and what you have here is the conductance of the root system. And then when the plant is transpiring during a given couple of days, what will happen is that during night you will have no transpiration, then during day you will increase in absolute value the leaf water potential, the soil will dry a little bit, then in the afternoon the transpiration will decrease, during the night no transpiration, the next day an increase of the transpiration again, it moves a little bit to the right because the soil is drying out, the Psi leaf becomes very negative during night, during day, and then again. Well, this is very nice, it's theoretical, but can we observe that? Yes, maybe a first point before that. This curve, we can, based on certain mathematical conditions, split it in two zones. One zone which is rather linear in green, and one zone which is rather non-linear in brown. And the green zone is controlled by plant. The dark zone, brown zone, is affected by soil. So the non-linearity in the system is brought by the soil, by the rhizosphere. And also a bit by the xylem when the soil is saturated. Can we measure this? Can we observe this? Yes. This is a system, I won't explain it into detail, but it is a system where you can measure transpiration versus the pressure. And here, I just translated the point there on my 3D surface. And you see that you can actually measure this 3D surface in reality. Okay, so now you are convinced that you can create models that represent soil and plant. Let's look a little bit more on data. Interesting. That's not what I wanted to show. Am I going in the right direction? Oh, it was. Okay, I don't know. So let's look at an experimental study that was done in Belgium on a vineyard. Very small scale. This is a field, 75 meters distance between this point and that point. Each of these subplots, we picked up 3 plants. On these plants, we measured the potential, we monitored the potential and the suffering, so the transpiration. At the same time, we measured water potential at different depths, water content at different depths. So we had the potential in the plant, actually in the leaf, in the trunk, and in the soil, together with the transpiration. And the nice thing with this plot, with this field, is that on the top of the plot, here, we have a lot of sand. So we have a small layer of loam and then sandy soil, while in the blue point there, you have only loam, so a fine textured soil. So it means that we have contrasted texture, contrasted soil between the orange and the blue, but the same species, wine, Chardonnay variety. So the biological aspect or the biological aspect of this plant or of the system is the same. The only thing that changes, the climate is the same also. The only aspect that changes is the soil. So when we do these measurements, this is a summary of the measurements. You had tensiometers, you had cyclometers to measure the water potential, you have subflow, and then from time to time we did LAI measurement and Schalander-Bomb measurement to have the pressure. And that's what we measured. So it was during a very dry summer. It happens from time to time in Belgium. And what you have on the top is the subflow, the transpiration of the system, and at the bottom, the potential in the plant. And this is the subflow as a function of the potential. So really see both going together. So what you see is somehow the heartbeat of the plant. You see every day an increase of the transpiration, you have noon and an increase during night. And the top value is not always the same because sometimes you have clouds, you have more wind, the demand changes. And here you have the potential of the plant. And what you can directly see is that the blue and the brown have a very different behavior. They have the same canopy, more or less. They have the same root system, but despite that, they have very different transpiration values. The blue is transpiring 3 times more than the brown. What you also see is that the potential is much more negative in the brown. Having a much more negative potential means that you have under more stress, you need more negative pressure to take out or extract a much lower amount of water. So it means that the brown plants have difficulties to extract the water, while the blue plants transpire a lot and have less water potential gradient. I like this kind of graph because you really see different behavior. You see also a bit of hysteresis, and you can easily make the link, maybe not so easily, but make the link between this and the 0D model that I showed you before. If you look at it from the leaf perspective, so you look at this graph from the right-hand side, this is the main slope, this is the root conductance, the root system conductance, and you are moving on this 3D surface. And what it means is that this 3D surface is not only plant dependent, but also soil dependent. Of course, if you have a measure of the bulb potential, of the trunk potential, of the canopy potential, you can also calculate conductances. It's easy, and we can check whether we have an evolution of the transpiration as a function of the xylem, of the soil water potential. You see differences between soil types, and also whether the conductances in the stem or below ground change with soil water potential. Here, you see no change of the stem conductance as a function of potential. It means that there is no cavitation. The only point where you see cavitation in drying there is this one. But the conductance of the system is very homogeneous. On the other hand, in the soil below ground, you see a lot of variation. When the soil dries out, you see that the conductance of the system is decreasing. This is an effect, maybe in the root, maybe in the soil. At this stage, we don't know yet. But then Louis did a simulation study where he could estimate that the soil in brown is generating a high resistance in the sand. You see that the brown line is below the green line. The green line is the plant conductance, and the soil is less conductive than the green line. While in the loam, the soil is in brown, plant is in green, and you see that the conductance of the soil, the rhizosphere conductance, is much higher than the one of the plant. It means that in the loam, the plant is driving the uptake. The plant is more conductive, is less conductive than in the soil, so the soil doesn't play a role. That's the amount of roots that you have and the ability of the plant to extract the water. While in the sand, the soil is controlling because the soil has a lower conductivity than the plant. And this is my last example. It's a global scale study that we just published, where what we try to do is to explain an observation that has been made by several scientists working on flux towers. Okay, so in that paper, what they did is that they took a network of flux towers over the globe. You probably know these flux towers that measure transpiration. And they took flux towers where soil moisture was measured. And when they did that globally on a very large amount of flux towers on different ecosystems, they always observed something that soil physicists and agronomists know, is that when the soil moisture decreases, the transpiration decreases. What they call evaporation fraction is the part of the energy that arises on Earth that is used to evapotranspire. And for all ecosystems, you always have this kind of behavior where you have a maximum transpiration when the soil is wet, and above a certain threshold that we call theta crit, the water content, the critical water content, you start to see a decrease. You can also see this as in this part you have an energy limited evapotranspiration. So this is the amount of light that controls evapotranspiration, while below this limit, water is limiting the evapotranspiration, so it's water limiting. And so we thought, okay, this value, what drives this value? What controls this value? Is it the plant? Is it the soil? Is it the climate? And are we able to predict this critical moisture content? So what we did is using our model. And of course, we also have a kind of critical estimation that we can find based on our model. What we define as a critical value is, okay, you see the surface. If you have a certain demand, atmospheric demand for evapotranspiration, let's say 5, this is the green line, so you have an original demand of 5. When this demand of 5 meets the line between the green and the brown, it means that to keep having this 5, the water potential in the plant should be very, very limited. And so to protect plants have a tendency to close the stomata. So our hypothesis is that stomata close in the limit, at the limit between the green and the brown clumps or surfaces. Sorry. And so we can always define for a given demand a critical soil water pressure head or leaf. And we did that, and we can, if we have a soil water potential, or pressure head, we can also find, thanks to retention curve, a soil moisture. So for any ecosystem, we can provide a certain transpiration, calculate this surface based on soil texture, and predict the critical soil moisture. And this works. So that's what we did here. So first, this is just to show you that the measured critical water content that you find over the globe are very related to the soil texture. If you have a more sandy soil, you have a lower theta crit. If you have a fine textured soil, you have a higher theta. So stress starts to occur at larger water. So the texture plays a big role for the value at which stomata will start closing. And this is the result of our model, where we have the simulation theta crit based on our model, and this is the measured theta crit based on flux tower and also subflow data. You see that we represent pretty well the average slope. So I think that the black line, continuous line, is a 1-1 line, and the dashed line is our model fit. Something that you can observe here as well is that you have very small variation in theta crit in sand, while you have a huge variation of theta crit in loam. We were also able to explain that. So this is the span, so the variation of theta crit. And here you have the sand fraction, and you see that we can actually show or explain. This is our model, I think. Remember, so each dot is our model. And yeah, each dot simulations, these dots are our model, and you have a fit, and you see that we are rather pretty well able to explain this fact. And what we can also do, because our model provides that, is to predict the soil water potential at which you have a stomatal closure. And it's interesting to observe that as a function of the soil type, the water potential at which the stomata will close will differ. If you have a very sandy soil, it will be less negative than if you have a loamy or a fine textured soil. Of course, there is also an impact of this at the global scale, because the theta crit varies as a function of the soil type, and if you have an increase of the potential level of transpiration in the future, the theta crit might change. And so if you are more in a sandy-like soil, your theta crit won't change much. If you are in a more clayey soil, the theta crit might change much more. That's it. So I wanted to convince you that below ground part of the plant is important, and that it plays a critical role in transpiration testing. I also tried to explain that we have different types of models, very explicit 3D models where you have all the root system, and very simplified models, and they have both the ability of giving us some information, very different one. 0D models are better at global scale, simplified model systems, and 3D models may be different. Of course, transpiration depends on climate design and plant and soil hydraulics, but we believe that soil is a main driver of the stomatal closure. And we can predict theta crit globally in all terrestrial ecosystems with this 0D model. That's it. Thank you very much for this opportunity to show my my studies. And this is a bit what we are doing. This is just for the story, a poster that my group did, where we were trying to invent new Tintin. You know, Tintin is a Belgian guy, huh? And we used ChatGPT to create fake covers of Tintin. And each title is a you see here, Red Wine Treasure. It's one adventure of Tintin. You also see Flight 7114 to Brazil. So we try to mimic cover without being exactly this. Okay, thank you very much for your attention.
Thank you, Mathieu. Well, I think you give preference for the present ones, if you have some questions.
I was thinking, how do you measure the water potential in the trunk continuously? Do you have an equipment to do
Yes, we have psychrometer.
Psychrometer.
So we use this ITC psychrometer. I think there was I don't know how I can stop the presentation. It's really bizarre. That's maybe not a good idea what I did, I'm sorry. But I can at least show it there. So it was shown on one slide here. So it looks like this. Okay, it's not very clear, but you have here you have a system that you push against the truck, and you can measure the truck water potential. And this system, you can use it also on a leaf and measure the leaf water potential for a long time. So it's well, it's pretty expensive. It's like 3,000 euro for one, but on trunk it works very, very well. You've seen the Nile graphs, and it's also very stable. So we had 3 replicates, and the 3 replicates were almost the same. So it was we were very happy. And to be honest, it's very difficult to measure leaf water potential with this system. It doesn't work well. But for trunk, it works pretty well.
I have one question. You talked about a very points model,
Yes.
and then we work in a larger scale, and then we run global models to predict climate change or impact of climate change in the plants. Do you think it's possible to find a way to upscale the equations, or you thought about the difficulty of the scale, but
Yeah, but for instance, here, a vineyard is relatively simple, but it's already a bit complex. It's not like a full cover, like a crop field. You have lines, interrows, so you have a certain heterogeneity. And despite this heterogeneity, we could represent this in 1D. So you can consider that you have several plants sharing the same soil and represent different sink terms within a given block of soil, even without representing them explicitly. So you're not obliged to have the 3D positioning of each root or each tree to be able to predict the system. But of course, you need to do some assumptions for this, like that all plants share the same soil. So it means that you have a tree, if you have three plants together on the same subplot, you will be obliged to say that if there is a root of one plant at 10 centimeter depth, all the other plants that have also root at 10 centimeters depth will be in competition with this first plant. I'm not saying it's bad, but I'm saying you need always to simplify things. And I mean, a lot of people do that. You're obliged to do that.
okay.
You know, in climate model, there is no roots.
Yes.
They don't consider root at all.
Yes.
So they don't consider
That's
the
right.
limitation of the transpiration. So the feedback between surface and atmosphere is somehow missing there, or not well simulated. So yeah, we can do better.
I'm just thinking. I'm not sure if it makes sense, but if you have a map for the soil, more sand or more lawn in an area, is it possible to predict
Yes.
the behavior of the plants? Because we are worried about the behavior of the plants in the future.
Right. That's what we tried with Louis. So
But in a map, especially
it's possible, yes.
but
But what I would do is probably take one plot or one part and the opposite, very different, and try to already see whether we expect differences between these two soil types. And then there are models working in 3D, but simplified.
okay. Okay, thank you. Any more questions?
I have many questions, but I'm not going.
I just have one last one.
Yes.
I guess. I was curious about that graph that you showed, that you have the sand and the land ends. The behavior of the transpiration with
Yes.
the type of soil.
This graph.
Yeah.
No? This one?
Yes, this one. Would it be the same plants planted in the two different types of soil?
Yes.
And can we account for different behaviors of plants? Like there are plants that are able to draw water even in drought situations. And
So when you say different plants, I mean, this was the same
same plant.
variety,
different-
same variety, but a different plant. You understand what I mean? When you say a plant is able to take more water,
Like they're more drought resistant, so they can draw more water.
so behind that, there are a lot of different options. So when you say it's able to be more resistant, it can be the fact that you can observe more negative water potential in the leaf, and that is really able to decrease this water potential to very negative value. It can also just be that it has a lot of roots, and that maybe the plant has some roots that are wetter soil, or that it develops a root system that is very efficient in going towards wet zones of the soils. So yes, we are able, but the problem is always how you parameterize that and how you get the information about it. I don't have a library when I can say, this plant, these are the parameters. No. It's still, you still need measurements. And I advocate that doing subflow and water potential measurement at the same time is really important to understand this behavior. If in this plot we had not measured the leaf water potential, we would have had a completely different view of what's happening. We could have interpreted things very differently. Like stomata was always closed for the scent, for instance, because 3 times less transpiration would mean they are always closed. It was not always the case.
Can I give kind of my view or interpretation
Yeah.
of and see if you agree? So, summary of what you said is that you can describe many of these processes in soil vegetation transfer processes, thus using physical laws. That you can also show that experimentally that many of these things really happen. I mean, you can really verify it. And that's now it's my vision now that the main difficulty to apply it, because you have experiments, you can say, okay, this is according to the theory. But now to predict it to a new situation, then you need to know some parameters of the new scenario. And among other things, you need to have detailed information many times about soil hydraulic properties and also about rooting characteristics. And I think the detail in which you need all that depends on the scale that you are working.
Exactly. So for instance, at global scale, obviously we had not the root.
You don't need
measurement.
details about root systems. But
Yeah.
if you want to simulate one plant in a pot, for example.
Yeah, we need a lot of details. Yeah.
So it's interesting, but important always to remember that the scale you're working on will affect the
Model you will use and the parameters.
What kind of information you need to run the models.
Yes, you are totally right.
Also, we agree.
Yeah, we agree.
Do you have any questions in the audience? No? Well, thank you very much for this very great presentation, and this model is really very pleasant.
You are welcome. Thank you very much.
And then I believe that there are other researchers here that may come back to you because they are developing
They can
models
do
for
it,
the
yes.
assignment. Thank you.
Thank you.
There are some comments on the chat. People are happy with talk.
Okay.
Thank you.
Okay. Good.